Hello, everyone. We're just going to pick up back from where we left off earlier this week due to the internet interruption that we had on campus. So, last thing that we were talking about was um, the four stages of mitosis. We were able to go through prophase, metaphase, anaphase, and telophase. And the last thing that I was able to say before being cut off is that cytokinesis, if it occurs, will happen towards the end of mitosis. Now, this statement means that cytokinesis does not need to happen in order for mitosis to be accomplished. It usually happens, but if it doesn't happen, mitosis is still able to occur as long as the other four stages do happen. In mitosis, what you start with is what you end with, and this is in terms of genetic material. So let's dig a little bit deeper. Mitosis is used by cells for growth and repair. It takes place only in eukaryotic cells, and in mitosis, duplicated chromosomes are separated into two identical cells. That is what starting with. That is what you start with is what you end with means. The number of chromosomes per cell stays the same, and they are identical in every which way. Going into the steps one by one, we begin with prophase, where the nuclear envelope disappears. So that is the first step. We all know that cells have a nucleus, but during prophase, the nucleus disappears. Genetic material tightens from chromatin to become chromosomes, and the mitotic spindle, made from microtubules, forms from the two centrosomes and attaches at the kinetochores. Now, at this point, you have to understand that the genetic material has already been duplicated in the S phase. So that we're not going to see anymore. At this point, we already have twice as much the genetic material that a cell needs. And that is because it's going to split it evenly among the two cells. Note, the microtubules from one centrosome connect to one side of the kinetochore, while the opposite centrosome's microtubules connect at the other side. The centrosomes serve to organize the microtubules and move towards opposite ends of the cell. As such, so you can see how the centrosomes begin growing those microtubules and how they approach the kinetochores of the different chromosomes at the same time the nuclear envelope disappears. Step 2, metaphase. Chromosomes are directed by the spindle microtubules to line up at the metaphase plate. This is an imaginary plane in between the spindles, very much like the equator. Okay? All chromosomes will line up on the metaphase plate during mitosis. And this is all being done by the spindle fibers. The spindle apparatus is moving these chromosomes towards the center, the region that we call the metaphase plate. Step 3 is when the chromosomes are pulled apart by the spindle apparatus towards opposite poles of the cell. The chromatids are now separated and are considered separate chromosomes. Remember, when we had the little X's like we were seeing, those were one chromosome. But when you separate them, now they become two chromosomes. So you should always remember that a chromosome that is duplicated and like attached at the centromere, it's a single chromosome, even though it has two chromatids. But once you separate those, each of the ones that used to be a chromatid now becomes a chromosome. So this is the point where we go from having 5 chromosomes to 10 chromosomes. As you can see here, they are further separated. So we had 5 chromosomes, they got separated, now we have 10 chromosomes. In the case of humans, we may have had 23 chromosomes, but now at this point there are 46 chromosomes. Telophase is the fourth phase. Nuclear envelope reforms around each set of new chromosomes, or not really new, but the ones that we already had and are now separated. The nucleus reforms around them, and the chromosomes uncoil back into their chromatin form, the uncondensed form. Now, this happens because DNA needs to be in that uncoiled form if it's going to do its function, which is transcribe some mRNA. Okay, in order for all the machinery to get within the DNA strand, in order to make that happen, uh, DNA cannot be that tightly condensed around chromosomes. Cytokinesis will usually take place as well, but again, I have to remind you that it does not need to happen. It doesn't have to happen, but it will usually happen at this point. And that is what you see here. The nuclear envelope reforms and the chromosomes virtually disappear because they go back into their uncondensed form chromatin. Cytokinesis is the separation of the cytoplasm of cells. In animal cells, a ring will form around the center of the cell, and a groove called a cleavage furrow will form as well. The cell will pinch into 2 new cells, as you can see here, and as you saw in the previous animation. Plant cell. A plant cell is synthesized between, a cell plate is synthesized between 2 cells when vesicles filled with cellulose move to the midline of the cell. So as you probably remember, plant cells have an additional structure in the outside that animal cells don't have. Plant cells have a cell wall, and a cell wall is uh, it would be very hard to just break it apart. Therefore, the newly formed cell plate gives rise to one side of the plant cell's wall, as you can see here. It works much like building a wall in between the 2 cells. Cytokinesis is an independent process that occurs only towards the end of mitosis. Here we have a breakdown of the whole process. We had interphase. This is where, after the end of interphase, we obviously went through the S phase, and now we have double the genetic material, as you can see here in prophase. Here we have duplicated chromosomes, whereas before in interphase we can assume that this was during the G1 phase. There was only enough genetic material for a single cell. Then it went into the S phase, it duplicated the genetic material. Remember, please, that that duplication does not happen while we have chromosomes. It is not like each one of these strands is just gonna grow a new strand. Uh, at the point of the S phase, we are in the chromatin stage, and each single DNA strand is duplicated differently, or individually, I should say. It is not until prophase that we actually get chromosomes, because this is when the nuclear envelope disappears and the chromosomes um, they tighten or they coil into that shape. In this picture we only show it this way just so that you understand that we have less genetic material. Now, in metaphase, they're going to line up along the metaphase plate, and in anaphase, they will be separated. In telophase, they will be pulled all the way apart, and the new nuclear envelopes will form. And cytokinesis may or may not happen at this point. So that's really all the material we have. Now let's just go over a couple of review questions. How many chromosomes are in this cell? The answer would be 4, even though we have 8 chromatids, okay? When chromosomes are attached in this fashion, they count as a single chromosome, not as 2 unified chromosomes, if you will. So we only have 4 chromosomes. Now, are they duplicated or unduplicated? The right answer here is that they are duplicated. What is the ploidy of this cell? This cell is diploid. How many chromatids are present in this cell? So we have 4 duplicated chromosomes, and each duplicated chromosome has 2 chromatids. Then we can easily say that we have 8 chromatids. How many chromosomes are present in this cell? We have 1, 2, and 3. We have 3 unduplicated chromosomes in this cell. What part of interphase does this process occur in? So we're talking about the DNA replication, the S phase. Now, again, I must remind you that DNA replication does not happen while we have chromosomes. It happens when we have chromatin. And it happens to each DNA molecule, okay? In the form of chromatin, not in the form of chromosomes. We do not see chromosomes until prophase. And that's it. Thank you for listening.